So the little funny design fail today is one where I really don't know how the designers of this truck could have done this any differently, but clearly you can see the ultimate end user, the customer who's using the truck, is maybe misinterpreting what the point of the holes in the hubcap here are, and that they're trying to tie this ATV down to, whoops, nope, there we go. Yep, trying to tie this ATV down to the bed of the truck, and you guys can sort of imagine that we're being set up for catastrophic failure here. Either the hubcap is going to fly off, or the belt, the tensioning belt is going to break, or the axle is going to bend, or the ATV is going to be crushed, or the bed of the truck is going to be crushed. One way or another, this is not going to end well. And I'm not sure quite how the designers of the truck could have changed the design to avoid the user deciding that the hubcap was a good place to tie this belt. But that is also something that we as design engineers have to think about and worry about. Is we design something that seems perfectly reasonable to us and seems to be really well designed, and then you hand it to a user, and the user uses it in some way that was completely unanticipated and wasn't thought about by any of the design engineering staff, and ends up either causing the thing to fail, or harming themselves, or breaking whatever the product is pretty quickly. And so this is just an example of that. So kind of a funny, another funny image here. Okay, so the thing that I want to kind of start out with is to congratulate you guys on surviving the RER. Almost all of you have now submitted the reverse engineering report. I have 111 submissions for 130 students enrolled. So that's really good. We did have a couple of kind of last minute snafus, technical difficulties last night, but I stayed up with the students that were having those and emailing me, and kind of work through them. So hopefully anybody that had any technical difficulties, we kind of have a path forward for you. But if you guys have talked to your colleagues and your peers who have taken this class before, the reverse engineering report is become somewhat of a rite of passage for MAE seniors. And now you guys have survived the test, right? So it's over, it's behind us, and I trust that you guys all did an amazing job. And I've looked at a couple of the reports, and the ones that I've seen have looked really good. So I'm looking forward to reviewing them and getting you guys feedback. I am going to assign them out to our graders and our TAs, and I've given them a 2-week window to grade them in. So today is the 30th. So you guys, barring any disasters or anything that I haven't foreseen, you guys should get feedback on your RERs by the 14th. So 2 weeks from now is when I expect the feedback to start coming back from the graders. So I'll let you know if we're going to miss that deadline for some reason, but I think that's pretty reasonable. There's about 11 or 12 that each individual has to read. Some of them are upwards of 100 pages, but I think 2 weeks is a reasonable amount of time to get them graded and get them back to you with some comments, which of course will be helpful as you move forward in your major project. So having put the RER behind us, we are now very squarely concentrated in moving forward with the major course project, which is where we're going to focus most of our time and energy for the remainder of the semester. And the major project, we've got it broken up into like 3 major reports, 3 major deliverables. And the idea there is so that you don't get overwhelmed at the end and feel like you have to write a 250-page report in the last week of class. We've taken like the big, hard, time-consuming, and we feel like there's 3 of them, big, hard, time-consuming aspects of the design process and put each one of those aspects in one of these separate reports. And then we've staggered the deadlines so that you're definitely going to be finished with the first hard thing before you have to start the second hard thing, and so forth. And these are all done in groups as well. So you guys have got now the ability to work with 6 or 7 other people to get these done. But the first one is the concept design report, the CDR, and I think I've posted the assignment prompt for that now. And the really big, hard, difficult thing in the CDR is creative ideation. So you guys remember from DML, you take a problem that you're trying to solve, you break it apart into subsystems or functions, and then you assign, or not assign, but you do assign, you end up assigning metrics to those functions, but you also then generate concepts, and those concepts are meant to be possible solutions to solve that problem or carry out that function of the subsystem of the overall thing that you're trying to design. And in this class, we ask you to generate 12 concepts per function or per subsystem. And the right number of functions is probably something like 6 to 8, given past experience with kind of projects of this scale in EML 4501. So if you've got, let's say 6 to 8 subsystems and you're generating 12 concepts per subsystem, that's, like 70, 80, 90, 100 concepts. It's a lot. And so that's why that is sort of the hard thing that we isolate from everything else and have you focus on in the CDR, and get that finished and wrapped up before moving forward into the rest of the design steps. So in order to do this creative ideation to generate these concepts, you guys have to know what the project is. And I've kind of broken from tradition a little bit this semester. And instead of waiting until the RER is done to reveal the project, because we have one less week in this semester than we have in a normal long semester, I gave you guys kind of what I described as a soft reveal, to share with you guys the customer needs statement last week, so that if you had time while you were taking breaks between your RERs, hopefully you reviewed the customer needs and you at least have a sense of what we're doing. Today, though, is meant to be the big formal official reveal where we would have the trumpets playing and the confetti flying and the ticker tape parade, and a big celebration moving forward of the next 10 weeks of your life, focused on solving this problem for our customer. And so what I've done is invited Robert Martinez, who you guys know. He's our graduate TA. He is also a PhD student in the laboratory of Professor Amor Menendez, who is one of our mechanical and aerospace engineering faculty. And Amor is a member of a consortium of faculty at UF. And faculty members in this consortium include people from mechanical engineering, electrical engineering, biology, medicine, bioengineering, and their ultimate overall goal, I think it's called the synthetic chemistry group, their overall goal, eventually moving forward, is to produce a biofoundry, called the UF Biofoundry, which doesn't yet exist at the University of Florida, but hopefully will. And one of the key things that they need to have a biofoundry is the microbio incubator that we did the soft reveal to kind of show you guys last week, and what Robert is going to talk about now. So Robert is sort of playing a dual role, right? Yes, he's our grad TA, but he's also our conduit to the customer. So the customer effectively being this group of faculty members that make up what will become the UF Biofoundry, using your design as one of the key components, of that Biofoundry capability. And so you guys really can think of Robert kind of as the voice of the customer. So if you have questions about the customer needs statement, if there's something that's unclear, if you're looking for more information on applications for this thing, how it's ultimately going to be used, all of those questions can be sent to Robert, and his primary job will be to answer them if he knows the answers, or take your questions up to the consortium of faculty and see if he can get a consistent answer from them, and then bring it back to you guys. So that's his role in this class, is to kind of serve as our liaison between this course and our customers. But with that, I'm going to stop sharing my screen and turn things over to Robert. And I need to activate share screen. There we go. So Robert, you should be able to share your screen now. And we will let him present on our major project.
Okay. Can you guys see the PowerPoint?
I can, yes.
Okay, cool. Awesome. Yeah, I'm doing off my tablet this time, so I can't get any feedback from watching the video. But thank you for the introduction, Dr. Traum. So as he had stated, we are currently in the works of trying to implement a biofoundry here at the University of Florida. There's only a few across the country, and UF wants to be one of the ones to have one. So with today's talk, we're going to go over the motivation. Why do we want to even go about doing such a project? The background, what prior work has been done already, a little bit about the project itself, and why the project's completion is significant. So the problem is biology is variable. So you can't avoid biological complexity. Characterization of synthetic biological systems are unreliable and tedious. Spontaneous physical variations happen with things such as different environments. And then you have something called evolution, which over time, you have things like bacteria who are able to have a generation time of like 10 minutes or less, which essentially means they can double their population within that time span. If there's any mutations along the way, it just precipitates throughout the later generations. So this, as you would understand, is a pretty big problem when it comes to quantifying these kind of trends in biology. So how would we go about solving these problems? Well, to go about doing this, we got to create a large-scale manufacturing, implement sensor monitoring, make it so it's repeatable, and then implement standardization. So as you can see with these medical scientists at GE, they have a large-scale version of a bioreactor, and we're going to make these but in a miniature scale. So with this miniature scale, they're going to be more modular in the sense that we can do smaller batches, so we can use this more for research purposes, where we could test thousands of variants in a time span of one experimental run versus running them multiple and if not hundreds of times. So what is the purpose of these bioreactor systems? So the purpose is that you want to create a continuous culture device that allows for the testing of, as I stated, hundreds of variants, control factors that impede growth, as well as tune those cultures so they produce the desirable outcomes that you want. So knowing that not many of you have biological backgrounds, some of these growth factors include our oxygenation, heating, what feedstock you're using, atmospheric pressure, as well as transcription proteins. So as Dr. Traum had stated, there's a consortium of synthetic biologists here on campus that want to come together and pretty much found this biofoundry to not only further their own research, but also make it capable so we can do a lot more research projects down the line. So that's where Dr. Traum and Dr. Nimi is a part of this group as well. These are just the scientists more on the biological side. So we got people from pharmaceutical sciences, the microbiology department, faculty from mechanical aerospace, chemical engineering, as well as plant sciences, all coming together for this project. So with that being said, the motivation, now we're going to get into what prior work has been done. So over in the University of Washington at Calvin's lab, they have their own turbidistat. For those of you who don't know, a turbidistat is basically a device that measures turbidity within a solution. So essentially you could think of it, how cloudy is a solution relates to how much of a given population of microbes are in that solution. So that's what they were able to build for under $2,000 for 8 units. And they were able to implement a PI controller for optical density and control temperature through use in an incubator. And you would see, if any of you guys have seen a biological or been inside of a biolab, incubator is essentially an enclosed environment that controls things such as temperature, humidity, and gases that are within the chamber itself to allow for optimal growth conditions. And that isn't really the best way to go about doing this, because if you got a lot of these little culture tubes essentially and all these wires hanging out, it could be quite cumbersome just to put them into a singular incubator. And then also the last point I put is that an asterisk, fluorescent detection. So that group was able to do a fluorescence intensity measurements on it, but it was on a separate project and it didn't do it simultaneously with optical density readings. Moving on, the Di Bernardo group out of Naples and Bristol, London, they also built their own version of a turbidostat, which cost about $200 for one of these units. And once again, similar to the Calvin's lab, it has a PI controller for optical density, temperature control through the incubator, but this one allows for multi-chamber versions. Good thing to, I guess, know if once you guys look into the literature, that these chambers themselves are also called sleeves because the vial itself gets slid into this sleeve-like contraption, which actually does the measurements. Next, what we're going to look at is the chat bio turbidostat. So this is when it gets a little bit more technically advanced. As you can see, the price tag reflects that in the sense that it's $6,200 for 8 units, but this one has not only fluorescent intensity measurements, but it also has the capability for a photosynthetic system. So it's saying that it can shine a light and grow cultures that require light to, I guess, subsist, such as like algae if you're wanting to do alternative biofuels. And then what's also nice about this particular device as temperature control per unit, and you no longer have to put it inside an incubator. And now we got the evolver from the Boston University over in Khalil's lab, I believe. He, once again, has his own version of the turbidostat. This one costs about $2,000. It's 16 of those sleeves. And this one has a PID controller, so now it has differential control in conjunction with the proportional and the integral control. And now, and this one also has, like the TriBio, has per unit temperature control, which is really ideal once again when you're trying to try a bunch of different variations for each of these individual cultures. So I have a list of resources because a lot of these projects are open source, so they have all their material available freely online. So you guys can check them out, see how they went about the design and build process themselves, and implement that towards this project. And here I have a, this is the Optron's liquid handler. And this, I have a video for you guys so you guys can watch to see how a device like this works. So apparently I can't play it off my tablet. So definitely that now that you guys have the PowerPoints, you should be able to watch this on your free time whenever you get the chance. But essentially what it just shows is, once again, it doesn't offer anything like the previous projects had in the terms of incubation or measurements, but it's able to dispense fluids in large capacities. Because once again, if you think of a 382 well, which is often used for measurement experiments in biology, manually pipetting 382 wells by hand is very tedious and very long, and it's prone to errors sometimes, given you know, human handling. And so what are we trying to do? We're trying to combine all these features. So we want something that has liquid dispensing along with incubation and measurements, adjustable working volume. So once again, in that video, they kind of showed a stand where they had these micro tubes in there and where they were doing a smaller scale experiments. We want to build upon that in the sense that we could use anything from 15 milliliter to 50 milliliter conical tubes, and then larger temperature range as well. So when I had mentioned with the evolver having a independent temperature control for each of the individual sleeves, it has a limited range from room temperature to 37, which is good for most, I guess, projects. But when you start going into other research endeavors, such as using extremophiles that like hotter temperatures, or even ones that like cooler temperatures, it also allows for other experiments to be done, such as DNA transformation, where you can do heat shocks now at 42 degrees Celsius, and also have cooling temperatures to at least 4 degrees, as I mentioned in the CNS, So you're able to slow, or if not inhibit, metabolic processes within the cells. And then lastly, what we want to do is add atmospheric conditions. Because once again, with this added capability, we could do a lot more in terms of research, in the sense that we could start emulating extreme environments such as those in Mars, where we can do high concentrations of carbon dioxide, we can introduce nitrogen, methane, and so you could only think of how many other projects we could do with these new conditions along. So with the customer needs statement, I'm not going to show the whole needs statement itself. I feel that's something it's better for you guys to read on your own. And if you have questions, definitely contact me. I'm widely available, as Dr. Trump said, either in my office hours or in the studio hours on Fridays, and also by email. So I'm just going to give you a general idea of how and why we broke it down the way we did. And so in the general breakdown, we did the general build features, which are like the safety components and the general mechanistic features that need to build such a device. The microbiological requirements, such as what do you need to, once again, bring up the cells, the components that deal with the cells, you want to make sure that they are biocompatible, in the sense that you can't use certain materials as they can kill the cells, or what's even more possible is having something that's porous, because once again, if you have any kind of cultures, they can get into those pores and subsist and further contaminate other wells, which we wouldn't want. And then you have something along the lines of measurements. So how are we going to induce this photosynthetic system? At what intensity? OD, fluorescence intensity, kind of like how we're going to go about doing these measurements. And then lastly, the liquid handling, where we it goes more into the lines of like at what kind of accuracy and precision we kind of would want from such a device. And then lastly, here at the end, if you guys noticed, or if you've looked into your customer needs statement, we had a try to have section. And I put that along the lines with considerations, because once with this kind of device, this is something that we want to not only be useful for our own biofoundry here at UF, but if other universities, if not high schools, want to like have such a device so that they can start teaching these skills to the next generations of engineers and scientists, they would be able to do so. So that's why we want something that is somewhere in the size of a standard bench top countertop space, as well as we wanted to focus on speed. I did say, I think 6.5 minutes for 682 well. That was just based off the dispensing rate I put in your liquid handling section. And that was just so, once again, like, we want you to try to do it as fast as possible without causing any black splash or volatility from any of the compounds getting out from the wells, kind of thing. And then we have ease of use and installation. So once again, we're trying, the center cornerstone of what we're trying to do is make that this is widely usable for not only our purposes, but if later down the line other researchers want to use a similar device as well, they can. So we want a device that is modular, in the sense that we could always add further additions down the line in a plug and play kind of fashion, so they can cater to the research that they're wanting to perform. And that goes along with installation. We don't want something that will take a whole group of people to assemble. It doesn't need to be so simple that it takes a day, but just essentially you could have one lab technician working in a lab be able to assemble such a device. So why is this significant? So just to reiterate, through large-scale manufacturing, sensor monitoring, repeatability, and standardization, we get things such as faster diagnostics. So as I stated, when it came to, let's say as an example with the COVID situation, we could do reverse transcriptase PCRs at a faster rate. We could do further detections to see if like cells are tumorous, cancerous or not. So it aids in that, and that we can see tests for 1,000 other outcomes, and that relates into innovation in the sense that we can try 1,000 different ways to combat a particular biological problem, and do it at a rate that is, I guess, better for everyone in general. And that goes along the lines with increased fidelity, in the sense that as I said, biological systems are inherently at the moment very variable. So how do we get something that works across all the lines? So just as you guys know, not all pain medications work for everyone in the same, because most medications are modeled after white males. So one pain medication may work for one person, but it doesn't necessarily mean it will even work on a female, as we have, as males and females have different pain pathways. And then that goes lastly into accessibility. So with such a device, as I had mentioned, we can be able to offer it to not only to the university level, but can offer it to high schools and give it to even, let's say, less privileged countries and such, and that they can start driving their own research to help their communities. And that goes even along the lines of like the pharmaceutical industry, where it's if any of you guys are diabetic or know someone who's diabetic, you know how expensive insulin has gotten lately. And little that you guys know is that it's pretty much ran by 3 pharmaceutical corporations, and they can charge whatever they want just because they have the information to do so. But when in all actuality, you pay $285 or so for a vial, it only takes about a few cents in the terms of the synthetic process to actually build that insulin product. So not only will you, as engineers, realize that you have the capabilities to change lives as you know it, and you guys are the means for that change. So that's why this is significant. And yeah, thank you guys for listening to the talk. I hope you enjoyed it. And if you have any questions, I'm free to take them.
Okay, great. Any questions for Robert? It's like Inception. I'm seeing like a copy of a copy of a copy of myself there on your screen. Okay, looks like no immediate questions. So I did manage to get the survey pushed out, and the survey has the same 2 what would well and what can improve questions. But then the third question is let me just read it here. What questions do you have for our design project customer who's with us that you did not ask? Oh, that you did not have an opportunity to ask. And so if you guys have a question that, you know, for whatever reason you're not wanting to ask now, you could drop that into the survey using, you know, just cell phone text, and I'll compile all those questions and I'll send them to Robert, and Robert can get us an answer. And again, if you guys are, you know, not wanting to ask a question now, but have them, Robert is available on Fridays, I think from 10:30 to 12:30. In the open virtual lab hours, and then he's also available on Tuesday afternoon starting at 4:30 for office hours. And you can always send him an email. So maybe Robert, if you wanted to just drop your email into the chat there. You guys can also grab his email from the website or the course syllabus. But one way or another, please, if you have questions, reach out to him. He again is there's the email. It's robert.martinez@ufl.edu. Remember the dot. Without the dot, there's a different person. I was emailing that poor person all summer trying to figure out, you know, why he wasn't responding to me. So that period is super important between Robert and Martinez. So yeah, one way or another, you know, if you have questions, reach out to Robert. His role in all of this, again, is to sort of serve as the liaison or the conduit between our class and the customer. And so to facilitate that back and forth discussion and dialogue and make sure that you guys have all your questions answered, that's his principal role as the graduate TA for the course. Okay, so if there are no questions, we can transition back